He's co-founded over 40 biotech ventures, one of which, Moderna, has helped change the course of a global pandemic. He has been referred to as the Edison of medicine and the father of modern drug delivery and tissue engineering. But behind all the accolades and patents is a relentless curiosity and drive to bring hope to patients around the world. I'm beyond excited to introduce Dr. Robert Langer, MIT Institute professor, a serial entrepreneur, and one of the greatest innovators in life sciences. Dr. Langer, welcome to Bioethics.
Thank you for having me.
So I wanted to start with some of your more formative years. You trained as a chemical engineer at the time when many of your peers actually went into petrochemicals. What pulled you towards biomedical research instead?
I was looking for a way that I could do something with my life that I thought would help people. I originally started trying to when I was a graduate student at MIT in the early 70s, I got involved in helping start a school for working class kids in Cambridge. And I was very interested in teaching and doing curriculum development. I tried to get some jobs in that area, but people didn't really write me back. And then I started to think of other ways that I could use my chemical engineering background to help people. And I thought about medicine. Most people didn't write me back there either, but finally one person did. So that's what I started doing.
Very cool. So early on, you joined Judah Folkman's lab, and the two of you were investigating angiogenesis inhibitors. At the time, many experts believed that delivering large molecules was just impossible. How did you muster the confidence to persist despite several funding rejections and a lot of skepticism from industry?
Yeah, we were going to solve the blood vessel problem, the angiogenesis problem. That was Dr. Folkman's vision. If you could stop blood vessels from growing, maybe that would be a way to treat cancer or other diseases. We had to solve the delivery problem. We had to have what we call a bioassay. I guess I just felt we had to solve it, and I wanted to. That was my job, so I just kept trying. And like I said, I failed hundreds of times. The grant rejections, those would come later, but this took care of my salary in the early years.
Could you walk us through that pivotal aha moment when you realized that porous polymers could revolutionize drug delivery?
I tried many different designs of polymers, lipids, other kinds of things, and either one of two things would happen. Either the drug would come out right away or would never come out. I always liked something visual. I had spectrophotometric assays. We were looking at large molecules, so I took certain enzymes. Different enzymes, they could do different things, and you could put them on like a microscope slide and have a gel around them. And depending on the enzyme that was actually coming out, it would cause a color change or something like that in the gel. So what I'd do is I would take these little nano microparticles, whatever, I'd incubate them in a solution like aqueous solution, and then I would take them out at different times, and either would change color right away and then never change it again over other time points, or it would just never do it. And so I would just try different designs, like different materials, different coatings, different concentrations of the molecule in it, and so forth. And every time one of those two things would happen. Finally, after I think maybe it was the 200th, I don't know exactly, it came out the first hour. But then when I came back the next day, it still came out. And it kept coming out for over 100 days. So that was, and the colors would all change. So, yeah, so that was basically, that it was just exciting to me to see that would happen.
And I think that's a pivotal moment that you're quite famous for. So moving more on to your work in brain cancer. One of your best known innovations is the gladiola wafer for brain cancer. It bypasses the blood-brain barrier and it reduces systemic toxicity. How did you and Dr. Henry Brem come up with this game-changing topic? Can you walk us through your thought processes and maybe some of the challenges that came along with it?
Sure. So basically, for us at MIT, it started trying to think about how to create an ideal biodegradable polymer. Going back to the 70s and 80s, there were really very few that were medical safe. And one of the things that we were interested in was coming up with one that would not only be safe and degrade into safe things, but that would show what we call surface erosion. It's like a bar of soap dissolving, surface first. And the idea was that the drug came out, it wouldn't dump out all at once. So anyhow, I had several postdocs, and graduate students, Bob Linhart, Cameron Young, Howie Rosen, and we would draw structures on the blackboard to try to think about what might do that. And then I got some other people that involved because I wanted to make sure that polymers would be strong. Gary Winnick was a professor at MIT then and gave us some help, and that it would be safe. And because you could pick different building blocks. And Mike Marletta, who now was at Berkeley, but then at MIT, helped us check those things out. So anyhow, it started with the structure, but we kept not just writing in the blackboard. We went ahead and we made it and showed that it did have these properties, at least in animals. So that probably started in 79, 80. Henry and Michael Colvin from Johns Hopkins came to see me in 1984. I'd known Henry going back to 1974 when he and I worked together with Dr. Folkman on these angiogenesis problems. But by now, he was starting his career at Hopkins as an assistant professor of neurosurgery. And they said, could we use that approach to create polymer that could locally deliver BCU? So we tried that and worked with Cam Young, and then actually went to Johns Hopkins and helped on it further. We got a company, Nova Pharmaceuticals, started by Sal Snyder, who's a very famous neuroscientist, and they gave us funding. And over the years, by everybody's work, we took it into early stage clinical trials and then phase 3 trials, and the FDA approved it in 1996, still used today.
Yeah, I wanted to touch on that translational aspect. These wafers have been around for decades now, but we don't see the widespread adoption of similar localized drug delivery implants in neuro-oncology. What do you think has been the biggest barrier? Regulatory, clinical, manufacturing, or just the science?
Well, I think everything. Part of it is what disease are you going to treat? Neurological treatments in general, unfortunately, there's a lot that's not understood. That gets to your point about science. And you would want to deliver it locally. I mean, that would be a requirement too. And you would want to do surgery. That would be important as well, right? It'd be better if you didn't have to do surgery. So those are all things that make that difficult. An example where the same idea is used and our patents were used for many years is actually in interventional cardiology. So the same principle is used in cardiology. Elazar Edelman, who was another one of my students, did a lot of that. But the point is, there, if you took Taxol, which is another anti-cancer drug, and that can prevent a problem called restenosis when you put the stent in. But if you gave the amount of Taxol orally, it probably would kill somebody. But you can deliver it locally by coating the stent with the polymer in Taxol. And when you deliver locally, it works quite well. And so that actually has had a major impact in revolutioning the whole stent industry. But I think the reason that works is because the science is more well understood, and they're actually putting the stent in anyhow. They're doing the surgery anyhow, which they're also doing in the case of brain cancer.
Got it. That makes a lot of sense. I now want to move on to some of your tissue engineering work. You and Jay Vasanti essentially jump-started modern tissue engineering, biodegradable scaffolds for growing new tissues and organs, and this was unheard of in the 1980s. What kind of sparked that leap from controlled release polymers to now building living tissues? Can you walk us through some of your experiences at the time, again, some of the challenges, and maybe how it was being perceived back then?
Sure. That was really Jay talking to me. What happened is, so I knew Jay also working in Dr. Folkman's lab. He was a surgical fellow, and we had published a paper together showing you could deliver anti-angiogenesis agents in mice. And we had done, it was a PNAS paper in 1980. Just like with Henry, I was good friends with Jay, so we talked. After Jay did that, he was a surgical fellow, and then he became head of the liver transplant unit at Boston Children's Hospital. You could see little babies dying of liver failure. And he came to see me when he said, Bob, could we come up with a way to create new livers from scratch? And we bounced around different ideas. people at that time had come up with some two-dimensional approaches for making tissues, but as we examined some of those approaches, you couldn't get enough cells in. And then Jay once I remember it was in Cape Cod and saw some seaweed and said, Bob, did you ever make a polymer like that? Because you change the surface to volume ratio and put a lot of cells in. So, I thought we could do that. So we did. And we worked together on it with a number of my students, like Saltzman and others, and showed that for the first time you could do something like that. But that doesn't mean it caught on right away. There was a lot of skepticism. And I think anytime you put an area like that, that kind of thing happens. But over the years, lots of great people, including David Mooney, who's now at Harvard, who's one of my early graduate students. He's did some great work on that, but a lot of people did. Now, Linda Griffith at MIT, Tony Nikos at Rice, all the wonderful students, they did a great job.
Yeah, absolutely a collaborative effort, but huge part on you as well. I wanted to ask, with the rise of organoids and lab-grown tissues, do you think that scaffold-based approaches will still remain dominant, or do you think we might eventually shift towards purely cell-derived architectures?
I think it may depend on the case. Just an interesting recent example that Laura Nicholson, who was another one of our fellows, then became the Yale professor and started a company called Lumicite, did. So when she was in our lab, published a paper in Science, she was first author in 1999. And so she made some really big advances, and we were able to make small diameter blood vessels using cells on polymer scaffold that actually based on some of David Mooney's work. At any rate, as she started the company, she realized that you could do it that way, but you could also decellularize the scaffold. Because once the cells did what they needed to do, you didn't need them anymore. So that actually just got FDA approved about a month ago. I think I just read something that's starting to get sold today. But that's a really exciting example. I don't think you'll see a one size fits all. I think right now skin is based on some polymer cell things. That's based on some polymer cell things where you get rid of the cells. I think that there's a number of other ones that can have different elements. I think you want to solve the problem with whatever tools you can in terms of understanding the biology, but you want to try to do it as simple as you can too.
Yeah, that's very cool to hear. I'm yet to hear of that, so I'll go check it out. So I think that's a really fantastic overview of some of your early work and groundbreaking achievements, collaborative effort, as you mentioned, but once again, a tremendous career you've had even just from that early stage. But I wanted to shift gears now and focus towards some of your more entrepreneurial ventures, specifically the biotech ventures and your story behind Moderna. You've co-founded and advised over 40 biotech startups. That's almost unheard of for an academic scientist. When did you realize that you wanted to build these companies yourself rather than wait for big pharma to kick up on the ideas?
What happened there? So when I did the drug delivery work, I'd worked on this for 10 years and I thought it was important. And yet nobody was really doing it. Finally, I got a patent. And then in 1983, I got a call from a very large animal health company, International Minerals and Chemicals. And they said, gee, we read your work. So we'd like you to consult for us. We'd like you to maybe give you a grant even. I told them about the patent. They licensed that. And I thought this was great because I thought finally something I did was going to maybe make a difference. The next year, I got another call from another very large company. And we made the same kind of arrangement. But what happened was each of those other companies, they did work on it for maybe a year or two, but then they gave up. They didn't get exactly the results they wanted. That was discouraging. And then maybe a year or two after that, Alex Klebanoff, one of my good friends at MIT and very well-known chemistry professor, said to me, Bob, maybe we could start our own company. So I was able to get those patents back, and we did. That would lead to a number of products. And it was, I think, a good experience overall. Students would get jobs. A lot of them were my students. And so we kept doing it and we continue to do it. I think it ended up being a good model to how to take laboratory things that are early and hopefully get them out to the world.
Yeah, I think that's a huge pull, or at least academic scientists. I know many who are being pulled more towards the biotech industry just because of that impact, but it also is challenging, especially if you're doing a startup, right?
Yes.
So,
What? It's challenging. Anytime you make a discovery and you want to then take the next step of getting it out to the world, you can publish things. And then, of course, we do that too. But to really put the time and effort to make a product, that's hard. And, and you get a lot of failures.
absolutely. And this is aside from the podcast, but I'm in a lab right now where we're doing CRISPR diagnostics for bacterial infections. And so we had the challenge of that academic research. You have to figure out the mechanism by which we can make this tool work. And then you have to get the tool to work, and we need to make it work consistently and get the numbers and the metrics right where we want it. And it's challenging because you have things often don't work in the lab, but then you also need to make it have performance metrics that can actually be used. And that is also challenging. And the patents, yeah, there's a lot more things to consider.
Sure.
But yeah, I wanted to talk about some of your earliest ventures like Enzotech and Neomorphics. Just what are some of the critical lessons that you learned from these early ventures that maybe you carried forward into your later companies? And yeah, what are some of the challenges and the successes? I realize I'm asking this question quite a bit, but I think it's important to get both sides of it.
Sure. Enzotech, I mentioned that, and we started the idea of making microspheres from both foods and drugs. And first of all, I didn't know what I was doing. And Alex is very smart, but I don't know that he necessarily did either. I remember one time he told me, wow, there's this guy I found who's like a senior director at a big company, multi-billion-dollar company, who's willing to leave his job and become our CEO. Geez, that's amazing, Alex. But later on, we found that he was getting fired from the job, and he was good, but he wasn't great. And I think the venture capitalists realized that. But we fortunately did get venture capital. And anyhow, we had a couple of things. In some ways, I thought this was great. We could use microspheres for foods and drugs, but it's hard to have investors value both. And that was complicated. So we all ended up splitting the company into two. And they both actually did all right. They didn't do great financially, but 4 or 5 of my students worked at them. The food company actually went public. They created some of the first fat substitutes that ever existed, and I think good sour cream and yogurts. The drug part, we merged with a company called Alchemase, and Alchemase is still around, it's done quite well. The microsphere type of technology, which they expanded over time, led to all kinds of products to treat schizophrenia, opioid addiction, and other diseases. And I think today is about 2,300 people. That's on the board of both companies up until 2000. They've continued to do a very good job. Neomorphics was a different model. Jay and I wanted to advance the tissue engineering. And of course, everybody said this is like science fiction. It could never work. But we did find some venture capitalists, Chen Weinberg and some others, and they gave us some funding. But we created it as more of a virtual company. The company would be virtual, but it would fund the lab. And we got some, I think, very good patents on it. And what happened is that one of the investors, Shriya, she became the chief executive officer. And the investors decided after about 4 or 5 years they wanted to sell it, but they sold it to another company that was doing some work. And the nice thing about that is that company would later make artificial skin based on this kind of work, which helped a lot of people, burn victims and people with diabetic skin ulcers. But also the investors got, I think, about a 3 or 4x return on their investment on us. And then that company gave Jay and me a grant for another 7 years. And we were on their scientific advisory board. So I felt that was a win-win too. Not a Moderna win or Alnylam win or some of the others that I've been involved in, but I felt everybody did well and got products out there that changed people's lives.
Yeah, I really wanted to touch on a point you made earlier, just about, convincing VC firms to invest in you. Early biotech seems to rely a lot on grant funding, like academic funds, NIH, DARPA, before the VCs even come in. And raising for biotech VCs is very different from traditional tech. It's capital intensive, long development timelines, obviously, and heavy regulatory risk. How do you convince investors to back a technology before it even has clinical validation? I'm asking you because a lot of students at Harvard do really have that entrepreneurial spirit. and have a life sciences bio background and eventually do want to create their own biotech startup. So it would be lovely to get your perspective on any advice or specific things that you've learned along your journey on how can you convince people to give you money.
Yeah. I think, first of all, people will give funding in biotech if they feel that you're going to form a transformative company, even if you haven't have clinical data. And the reason is that that's happened a number of times. I remember the first company I consulted for, Genentech, when they were starting this idea of what was then called genetic engineering. It was the earliest work in the 70s. And it was almost like revolutionary. There was so much excitement that you could actually really make drugs with genes. And I remember, I think the day that Genentech came out on the stock market, and they were nowhere near having anything. And so the stock tripled in the same day. And I think there's a story actually that I'll mention a book that your colleagues might like, but and I don't hope I'll get the book right, but there's a story, which is a true story, that apparently there was a young student, who was a high school student, and he wanted to have a summer job at Genentech. And he went there and they said, we don't have any money, but we'll give you a little stock. And he got the stock. And when Genentech went public, he made $4 million. This was 1970s. And that was because the stock was worth so much. I think the book, and it may be in the book, regardless, this book is a good book named called Invisible Frontiers. And it is the race for the insulin gene. It talks about Genentech and Biogen and Cairo. At any rate, and there's so many examples of that. And the reason for that is there's sales, but there's also value. And the way I would look at it is a lot of companies that don't have these regulatory requirements, what happens is if they have a lot of sales, they'll get a certain valuation. Valuation may even be less than the sales. That's not the case in biotech. In biotech, you can have no sales and huge valuations. And that happened with Genentech. And I'll give you a personal example that one of the companies I helped start is a company called PureTech with Daphne Zohar. And they spun out a company called Karuna. And Karuna came up with actually a new treatment for schizophrenia. And Bristol Myers bought them for $14 billion. And they did not at that time have FDA approval. But they felt the promise of this was such that it would be well worth it. And I believe that will come to pass. Now it's FDA and stuff like that. Value is key. So how do you convince the investors that you have value? I think you need a big idea. You do need patents, and you do need good animal data, I think, or at least good data that will convince them. But those are some of the elements that we've usually done. And that's why, like, for example, when you're talking about CRISPR that you're doing, well, there's a lot of hullabaloo over patent issues because different companies would be talking about that. But I think those are some of the elements. And you need good scientists, you need good everything, right? But it's happened many times, not just to me.
Absolutely. I think like biotech startups, there's huge technical risk. Is this going to work? Is this going to make it through the entire pipeline versus tech companies or like SaaS companies? It's more of a market risk is what I've heard from some people having that data that no, it works. I wanted to talk about Moderna, founded in 2010. mRNA was still considered high risk, unproven science kind of at the time. What made you confident that mRNA-based therapeutics could become a platform for vaccines?
I don't know that you ever know these things for sure, but Derek Rossi had come to see me. He's a professor at Harvard, and he had done some, I thought, very interesting work showing that if you modified RNA, you get maybe what I call the interferon response that would make people sick. And that was built on the work of people at Penn, Tomiko and Weissman. But I didn't know that then. All I knew was he basically showed me very nicely that he didn't get that response and he was interested in starting a company. I think there's two issues on maybe making things successful. One is that, the other, of course, is delivery. And I certainly felt by that time I'd spent 36 years working on delivery, that was something we could probably help on. And he was a very smart guy. We also got terrific business people like Nubar Fayan, I was on his thesis committee, he started Flagship Ventures. but he was interested and actually did an amazing job. And Ken Chen was a cardiologist. So that was the four of us that would start it. And later on, we've got outstanding people like Stefan Vassell, who's the CEO, Stephen Hogue, who's and still are the president, Melissa Moore, who's just a wonderful scientist. So it was, again, we had certain elements. We did write certain patents. We had great people. And when the data would start to look good, first in small animals and then larger animals.
Makes sense. So I want to fast forward to the COVID-19 pandemic. Obviously, Moderna's vaccine was among the first authorized. How much do you think it was a right place, right time sort of thing? And how much was the culmination of decades of research in macromolecule discovery?
I think it's both, but I certainly felt, and I think investors did too, and certainly the company, that Moderna was going to be very successful no matter what. Except for COVID, of course. But at that time, Moderna was in 13 clinical trials for both vaccines and for molecules. Now it's many more. But I think that we certainly felt that you could use this technology to solve big problems, whether it would be other vaccines, new ways of treating cancer, other diseases. But sure, the fact that COVID came along and nobody else other than this one little company in Germany, Bioethics, was doing much, certainly that made a giant difference. And it was very effective technology, far more effective than standard ways of making vaccines. So I think it would have happened anyhow. It just would have happened later.
Absolutely. I want to mention and just talk about the pace at which Moderna scaled. Grew from a small startup to a global powerhouse, distributing hundreds and millions of vaccine doses. What were some of the biggest challenges in scaling so fast, scientifically, logistically, and maybe even culturally?
Yeah, the biggest issue, if you ask Stefan, who was the CEO, was probably manufacturing. Fortunately, that Moderna had built, and we remember we discussed this at the board of directors, a manufacturing plant in Norwood, that, and we did that for other vaccines. But we had that, but still manufacturing probably was the biggest challenge to shift that over to do this. And so that was one challenge. People, and probably the biggest. In fact, Stefan sometimes says if he had to do over again, what would he do? He said, probably I should have put more money into manufacturing. Of course, problem is you never know. And when we put $100 million plus into manufacturing for a tiny company, you get criticized a lot already. But at any rate, that was probably the biggest. Everything was a challenge. The science, of course, then COVID hit. The people at Moderna were super dedicated. They worked around the clock. They did an incredible job in terms of people's morale. When you have the whole world watching, actually some portion of the world protesting too outside the Moderna buildings because they felt Moderna should give it away for free and things like that. And you're dealing with everybody, reporters, stock analysts, the press, and, but mostly people's lives. And so you just want to do the best they can. But I think it was an incredible pressure cooker, but the people that worked at Moderna and that continue to work at Moderna, they were amazing.
Yeah, absolutely. Now, with the few minutes we have left, I wanted to shift more towards looking forward and the future of science, biotech, and medicine. Obviously, your controlled release technologies have really paved the way for biologics, but now we're looking at gene therapies, CRISPR, siRNA, mRNA. And I want to talk about delivery. Where do you think is the most promising advance in delivering gene therapies? Do you think it's lipid nanoparticles, viral methods, or something else entirely?
I don't think there's a one size fits all. And I think it's an area of continued research, lipid nanoparticles and other kinds of nanoparticles, whether they be polymers or carbohydrates. I don't know that the perfect nanoparticle by any means has been created. And I don't know that the perfect vector has been created either. I still think lots of things can be done and need to be done. It's great. Lots of people are doing it. And I think we'll continue to see more advances. But and I don't know that we'll ever see a one size fits all. The challenge for delivering something like a DNA is different than RNA. And the challenge for delivering siRNA is different than delivering messenger RNA. I mean, why is that? RNA to a DNA, you want to get into the nucleus. RNA, you don't need to do that. In a way, it's a much different problem. Why is siRNA maybe different than mRNA? It's a smaller molecule. So there might be chemical approaches that you could use. And Alnylam, for example, has done a very good job on actually both those things, lipid nanoparticles, but various chemical approaches. And there's other companies too. But it's just inherently different. So it would be a mistake to look at a one size fits all. CRISPR, a lot of times what's being done is to do things outside the body. It's just much easier. But of course, that's less convenient for the patient and may limit what you do. Someday, people will be able to do it inside the body, but advances keep happening and happening. That's why we need more and more funding for both basic science, Harvard and MIT and lots of other places, but also for our companies that do these things.
Yeah, I absolutely agree. And that's actually one of the last things that I wanted to talk about is the idea of funding research. Some of your early work almost didn't get funded. And if agencies don't back these high risk ideas, we may lose out on a lot of big breakthroughs. Do you mind briefly speaking on what you would like policymakers and funding bodies to know about supporting radical innovation? Because I know there are lots of studies being published where scientists are more likely to submit grants with, let's say, lower risk ideas or ideas that they believe are more likely to get funded. And obviously, no risk, no rewards. I just wanted to get your perspective on that. And also, if you don't mind speaking to like the current kind of landscape of funding academic research and where you see that going.
I think there's different things, academics and companies and so forth. For academics, I do feel that the government in different ways, NIH, DARPA, ARPA-H, places like that, I think that's very important that they fund high risk things. Lots of times when money's tight, you have study sections, which are usually many professors. It's difficult to people to fund high risk things. I think it's always important to try to encourage reviewers and study sections and administrators to ask what's the benefit, not just why something won't work, but what would be the benefit if it does work. But there's no easy answers. I think that those different funding places have different goals, and maybe they should. We do a lot, for example, with Bill Gates, and he has very different goals, say, than the NIH, but all of it is aimed at doing good. So I think getting funding from wherever you can is important in the foundations. And so I hope that that happens. If you're a young professor or an old professor, you just have to hang in there and keep trying. And you may get a lot more no's than you get yeses. It's okay. I think you have to keep trying, even if it's discouraging. In terms of the current situation, I think it's not clear yet. And I think we have to see what happens. There's all kinds of lawsuits and everything else. And I think I just don't know yet what's going to happen. Obviously, I'd like to see more money than less to support research. And I'd like to see more money than less to support companies too.
Absolutely. That is the last question I had. Maybe if you just had a few words of advice to our audience is mostly Harvard and MIT undergraduate students. So if you had maybe a few words of advice to students who are wanting to go into industry or maybe start their own company.
I think in biotech, you want a big idea. I think you want to not start it right away. I think we do something to practice. I think you do want to have a good patent. I think you want to have a great team. There's a lot of things that you want to get, but those are some. And it's experience of no matter what happens.
Absolutely. Thank you. Thank you so much for your time, Dr. Langer. It was just truly an honor to be able to speak to you. And I hope your cold goes away soon. Enjoy the rest of your evening. Have a good one.
Thank you. Bye-bye.